Welcome to our overview of bioenergetics and oxidative phosphorylation, the fundamental processes powering cellular activity. In this section, we explore how energy-rich molecules are metabolized to drive the synthesis of ATP, the energy currency of the cell. Energy-rich molecules like glucose undergo a series of oxidation reactions, ultimately yielding carbon dioxide and water as byproducts. Metabolic intermediates donate electrons to coenzymes NAD+ and FAD, forming the reduced energy-rich carriers NADH and FADH2. Oxidative phosphorylation occurs continuously within the mitochondrion, the site where electron transport and ATP synthesis are coupled. The mitochondrion features a highly permeable outer membrane and a specialized protein-rich inner membrane essential for OXPHOS. Cristae greatly increase the surface area of the inner membrane, while the matrix hosts enzymes for the TCA cycle and other key pathways. The electron transport chain acts as the final common pathway for electrons derived from various metabolic fuels. 5 separate protein complexes, I through V, are embedded in the inner membrane to facilitate electron transfer. Coenzyme Q and cytochrome C serve as mobile electron carriers, shuttling electrons between the fixed protein complexes. As electrons flow through the etc., they lose free energy, which is harnessed to pump protons across the membrane, creating a vital proton gradient. Let's examine the specific molecular mechanisms occurring at each protein complex within the respiratory chain. At complex I, NADH donates electrons to flavin mononucleotide, initiating the flow of electrons that drives proton pumping. Complex II facilitates the transfer of electrons from succinate to coenzyme Q, bypassing the proton pumping steps associated with complex I. Cytochrome oxidase, or complex IV, is where electrons, protons, and oxygen combine to form water, completing the respiratory process. Inhibitors can bind to components of the chain, blocking oxidation-reduction reactions and halting the production of ATP. Understanding the thermodynamics of these reactions is key to grasping how cells maintain their energy balance. Redox pairs are defined by their standard reduction potential, which predicts the spontaneous flow of electrons through the system. The change in standard free energy, or delta G, is directly related to the magnitude of the change in reduction potential during transfer. The coupling of electron transport to ATP synthesis is highly efficient, though some energy is released as heat to maintain biological function. In summary, oxidative phosphorylation is the essential pathway that transforms metabolic energy into ATP, supporting all cellular life. Thank you for joining this exploration of bioenergetics. Understanding these processes is central to mastering cellular biology.